Excretory system has two major roles in an organism. First, maintenance of homeostasis, that is being balanced between the internal and external surroundings. Second, the removal of waste materials produced by the cells as a result of numerous metabolic activities such as nitrogenous waste, which is not removed can lead to the poisoning and ultimately leading to the death of the organism. Thus, the principal excretory product in gaseous is CO2 or carbon dioxide, liquid is honeydew, solid is urea or uric acid, semi-solid is allantoin. Moreover, crustacean arachnids possess paired excretory organs, maxillary antennal causal glands that open at the bases of certain appendages. Myriapods, insects, and some arachnids such as spider and mites possess another type of excretory organ, mammalian tubes which open into the intestine. Thus, these animals, both excretory and digestive waste exit from the anus. Three types of excretory organs are found in the acarina. The most primitive is the costal gland. Excretory tubules that open into the hindgut but which are endodermal in origin are the most usual type. Hindgut of the Trumbidiforms becomes modified to form an excretory organs. This image is an example of excretory system in acarite. In addition to these organs, the cells of the midgut are excretory in function. During digestion, the cell becomes filled with excretory products that are cast off into the lumen of the midgut from whence they pass into the intestines. Mites excrete their nitrogenous waste in the form of guanine. The Chondridiforms have no excretory tubules of usual type, but the hindgut is modified as an excretory organ in many of them. Causal glands are present in some of the Chondridiforms. The nostostigmata have two excretory tubules and a pair of causal glands that open on the coccyx 1 or 2. The Sarcoptiforms have a pair of small excretory tubules. Causal glands have been found in the Oribatids. The Holotyridae have two pair of excretory tubules and a pair of causal glands that open on the coccyx 1. Modeling the Malpighian tubules of a predatory mite. Malpighian tubules. The main organ of excretory and osmoregulation in arachnids are the Malpighian tubules, acting in the association with rectum and ileum. Malpighian tubules are outgrowths of alimentary canal and consist of long thin tubules formed in a single layer of cells. Each tubule is externally covered by peritoneal coat and supplied with muscle fibers and trachelius. Functional differentiation of tubules was seen with the distal secretion region and proximal absorptive region. Other excretory organs. Nephrocyte. Cells that sieve the helminth for products that they are metabolized, or precardial cells. Fat bodies. A loose or compact aggregation of cells, mostly trophocyte, suspended in the hemosol responsible for storage and excretion. Next is oinocytes, the cells of hemocoat, epidermis or fat body with many functions. Integument, the outer covering of the living tissues of an arachnids or mites. Tracheal system, the arachnids or mites gas exchange system, comprising trachea and tracheoles, and rectum, the posterior part of the hindgut. Among the above organs, Malpighian tubules are the major organs of excretion. These are excretory structure of few arthropods.